האמת הזאת כזאת יצאה לקרוא ממנה, נראה לי כאילו, אבל בסדר, תשאר פה, כן. אני רוצה לקרוא פעם מאוד, אבל כמעט נפלתי משם, כן. עד שעה, אז די גמישים לזה, אחרי שעה אנשים כולל של זה לפנף לי אחרי 45 דקות. 5? תודה רבה. Okay, welcome everyone to the last seminar of the semester. Today we have with us Elad Schneidman from Weizmann Institute. He's a theoretical neuroscientist from the Department of Brain Sciences, and he's the head of the department. He studied the interface of neuroscience, computation, machine learning, and statistical physics. How large groups of neurons work collectively to encode the process of formation. He also had both experimental and theoretical works, learning in memory in humans and in animals, collective behaviors and learning in other groups, in human and other groups of animal repetition agents, and architecture function relation. He got quite a few awards during his career, including Gunther Young Scientist Award, Rockwell Award, Wolf Foundation, and so forth. Thank you for being here.
What ChatGPT tells you about people. Yeah, you should never trust what the ChatGPT tells you about people. So hi everybody, it's kind of always wonderful to be here. It's kind of a homecoming back to where it all started, and it kind of raised a lot of wonderful memories, so it's great to be here, although I've never been in this building as a student, but still. Good, so my group works on a range of theoretical issues, and I want to talk about, I was kind of going back and forth about the stuff I should tell you about, and I'm going to just mention briefly the stuff that I'm not going to talk about in case you find it interesting, so you can chat with me about this later. We're spent a lot of time thinking about and working on collective behavior in groups of fish and in groups of mice and groups of networks and artificial agents. So there's kind of a bunch of stuff that we did in terms of how collective behavior or interesting collective behavior emerges as a function of the interactions between elements. And in particular, we're interested in simple things that control or result in complex behavior. And the other stuff I'm going to maybe touch at the end, depending on the time and how things flow. We've been playing a lot with connectomes these days, trying to come up with what you can think of as design features of how maps of connectivity are built and what they actually do. So I'll try to get some notion of this at the end, but we can talk about this later if you want. What I'm going to talk about mostly today is about neural activity, and this is a movie I stole from Takashi Kawashima, but you've seen many of these. This is kind of imaging of a larval zebrafish with calcium indicators in the neurons. I wish this was the real colors that they shine in the experiment, but this is, of course, fake colors to try and reflect different kinds of neurons acting together. And what we'd all like to be able to do is to look at one of these activity maps and read what's going on. So this is kind of a similar kind of data, but comes from this case the prefrontal cortex of a monkey watching moving dots and making decisions. And so you've seen these many times before, I'm sure. This is a matrix that kind of describes the activity. There's 100 neurons here. Every line in this matrix is one neuron. We put a dot whenever there was a spike, and there's a little over 2 minutes here in terms of activity of this particular part of the brain. And what we would like to be able to do is to be able to read this, right? But until we get our very own Rosetta Stone, we need to make sense of what's going on in some other ways. And this is hard for multiple reasons, but maybe the dominant one, which I'm going to talk about today, is the fact that is the magnitude of this problem. So if you think about the activity of a population, if you even take a small snippet in time, and you just ask about the binary word, if you will, that describes the activity. If you think about 100 neurons, the number of possible patterns is painfully big. And so any attempt of trying to make sense of the rules of this language and how it's built is not about how long of an experiment you run or how accurate you do it. This is really about trying to find some kind of simplifying principles that govern what's going on. And so what I want to try and do today is to talk about the rules that we think seem to be governing the joint activity of large populations of neurons in different parts of the brain. In particular, I'm going to talk about the cortex. Then I'm going to try and ask how we can actually learn these codes well enough to understand what they do and how to maybe even interfere or play with them. And also something about how the brain might actually learn to do that. And the starting point for this is to ask, if you think about the joint activity, kind of a fundamental thing about this is, how much of correlated or joint activity is there that we should worry about. So if you kind of zoom in on some small part of the data, and you just, this is kind of a blow up right here on the right. You see the marker here, I guess, right? Which is only in my, it's there, fine. So if this is a zoom of the activity, and you just think about the binary words that describe what 10 neurons are doing in different time bins, where every time bin here will be 20 milliseconds or so, the fundamental question about how you should look at this is really how correlated are the neurons as a group. Can you, what do you lose by ignoring or looking only at what individual neurons do, which is the common version of what you do when you look at firing rates, for example. So now this feels like a kind of a simple question if you look at small groups of cells, and the answer for small groups is that it seems like correlations are relatively weak. These are two examples. On the left is just a summary of the correlation coefficients between neurons. This particular data set comes from the retina, but this has been seen in many different places. You look at the typical correlations between neurons. The typical correlations between them is relatively weak, on the order of 0.0 something, right? Not something you write home about. And if you try to ask about things which are more complicated in terms, if you look at pairs of neurons and you ask, okay, maybe their correlations is weak, but something about the information that they carry is interesting or magical. So this is, again, a summary for the retina. This is over many, many experiments in Michael Berry's lab many years ago. And so every dot in this plot is one pair of neurons. The x-axis is how far they are on the retina. The color of the dot is the kind of movie that the retina was watching in this experiment. And the y-axis describes the redundancy between neurons in terms of how much information they actually carry about the movie, where 1 would be perfect redundancy, that they replicate to one another. -1 would be perfect synergy, which means that you only have something at the level of the pair that you don't see from individual neurons. And while there are neurons that pairs that seem to be redundant to a significant degree, most of what's going on is very close to zero. Namely, the neurons are not very far from being independent. And so with that thing in mind, you kind of have the notion that maybe this goes back to the old idea of redundancy reduction, that neurons are, the brain is built in a way to try and maximize the information that neural circuits carry about the world. And so to do that, you get rid of redundancy and you create an efficient code. This is kind of go back to the old ideas from Barlow and Atnev. And of course, nobody was insulted with the notion that the brain is optimal, it turns out. But this very nice paper that, if you haven't seen it, is kind of Barlow himself 40 years after the first one, in which he said, this idea is kind of weird because if you got rid of the redundancy, you become very susceptible to noise. That's problem number one, and we know the brain has a lot of noise in it. And the other is that our ability to learn from examples comes exactly from the fact that there are correlations or structures in terms of how neurons are related to one another. So it turns out that if you look at even relatively small groups of neurons, not 2 or 3, but already at the level of 10 neurons, something else completely jumps at you. And this is, again, data from the retina. I'll get to the cortex in a few minutes. But this is a summary from one of the experiments back in Michael Berry's lab in Princeton. And this is a summary of 1 hour long experiment. Every dot in this plot is one activity pattern that we've seen over the response of the retina to the movie. And so I'm just marking 3 examples. So this will be the first, third, fourth, seventh, and ninth neuron firing. So the x-axis is how often you see this particular word or pattern in the experiment. And the y-axis at the moment is a model that predicts which patterns you will see if you only rely on the firing rates of the neurons. So you ignore these seemingly weak correlations between them. And you can, and the black line over here is equality, and this makes orders of magnitude mistakes in terms of predicting what's going on. And so to try and make sense of this, or ask what's going on in terms of actually creating these correlations, we went back to an old idea we stole from statistical physics, which is to say, let's build the minimal model that takes into account all these weak correlations that we have. And the way to do this is to ask what's the most random or least structured model that describes the data that obeys these weak correlations but doesn't assume anything else. And this is the idea of maximum entropy distributions. So technically what we do is we try to optimize or to find a probability distribution, which I'll call p hat. So this is our prediction of which patterns will appear in the experiment, where I require that the model will give the same firing rates as what you find in the data. So this will be controlled by this term over here. And I want also it will capture the pairwise correlations between them, which is this term over here. And if you try and maximize this thing, you get the most random distribution that's consistent with the firing rates and correlations, but no other structure, which is what James proved in 1957. And what's nice about this is that this gives a unique answer. The problem is a convex optimization problem, so we know there's only one, and we can actually get there numerically. There's no analytical answer, but we know what the form of the solution is. And this means that the probability of observing particular patterns will be an exponential distribution where there'll be, if you think of the activity as x1 to xn being binary variables describing each of the neurons being active or silent, this will be an exponential distribution where you need to learn one parameter for every neuron and one parameter for every pair. So you end up with something which is in the order of n squared parameters. And this works really well. So this is the same data from before. The blue dots are, again, the prediction of the model that ignored these correlations. The red dots are what comes when you you build the minimal model that's consistent with these correlations. And so this maximum entropy model basically gets things right on the line. If you're worried about the scatter over here, you don't need to, because this is really within the error bars of actually measuring things in the experiment. So this is as good as it can possibly get. So even at the level of 10 neurons, we see this collective behavior that emerges from all these weak dependency between the neurons and govern what's going on really well. And since we've done this many years ago now, this has been seen in many, many different systems. However, if you go up to larger populations, things start to break down a little bit. So this is now recordings from 100 ganglion cells in the retina. Every dot, again, is one of these binary patterns. Again, x-axis is how often you see it. Y-axis is the predictions of the models. The gray dots are, again, predictions of the model that ignores the correlation. The pairwise model is the dots in red, and you see things look pretty good, but something is off in terms of actually the most common patterns in the experiment. And things look even worse if you try to do the same thing looking at temporal patterns of the neurons. So the pairwise model seems to be doing a good job for small groups of cells, but something is starting to fray off when you look at around 100 cells or so. And so this presented a very depressing mathematical notion of how should you try and describe the code of such large populations, because if pairs are not enough anymore, and the beauty of this formalism is that this is the most juice you can get from all the pairwise relations. That's the power of maximizing the entropy here, that if I need triplets or quadruplets and so forth, then I need to take into account the fact that the number of triplets and quadruplets grows in a rather painful manner. So there's no way I can do this for even 100 neurons, because if I need all the triplets, that's 100,000 triplets, and 10 billion quadruplets, that's really not going to work. And so what we noticed is that when we looked at the data, kind of trying to look for something that will save us, is that if you look at the distribution of the patterns themselves, and you do what linguists do when they collect words in text, there's kind of a zip-like law in terms of how the words appear. So this is now reordering. Every dot here is one particular pattern of activity, but I'm ordering them according to how often you see them in the experiment. And you see there's kind of a parallel tail here that describes what's going on. And so we thought maybe we don't need all the triplets and quadruplets and so forth. Maybe we can just use the most common pattern that we see in the experiment to try and learn something about higher order terms that are really important. And so to cut a long story short, this is kind of the prelude to what I want to show you. We found a way to identify a relatively small number of higher order terms that seems to be doing a really, really good job. And what's even nicer is that we need a relatively sparse set of high order terms to actually try and describe the code book of the system. So kind of something which is close to order n terms. So we need a lot of the pairs, not all of them, a handful, a few tens or hundreds of triplets depending on the population, and a handful of quadruplets. And this results in super accurate models. So this is now a bit of a different way to show you the data, but to show you how well things work. Again, every dot is one pattern. This is now for groups of 70 neurons, so we can sample the data probability well enough. So this is how often you see it in the experiment. And now the y-axis measures the agreement between the model and the data. So this is log likelihood ratio between the model and the data. 0 would mean that you have a perfect prediction. This blue funnel here is what you get just from sampling noise, so everything within the funnel is kosher, so to speak. So the independent model, we know that ignore correlation sucks, but this is just for a reference. The pairwise model is missing mainly the most common pattern in the experiment, and this is what happens with this thing that we call the reliable interaction model, where we identify a handful of high-order terms that, and this is all, of course, cross-validated on data that we did not train on. And it does give a nicer job when we look at temporal patterns. So these models are really accurate in describing which patterns you'll see at the level of predicting individual patterns with an accuracy that is as good as doing the experiment again. Okay? So it seems like we have a way to try and describe, if this is the kind of population you want to play with, we have a way to learn the rules of this vocabulary really, really accurately. And there's a small set, something which is not very far from order n, or maybe n log n number of parameters that you need to describe the vocabulary of the code. So we've done 10, we went to 100, and then so what if I want to do more, right? And then it's clear that none of the things I described to you can actually work the way I described them, because, maybe you should have been worried already by seeing that for pairs, it seems like you're not missing much by ignoring correlations. For tens of neurons, you need pairwise terms. For 100 neurons, you need higher order terms. So it's not clear that you should expect that things will look similar for more cells. But it's not clear how to expand any of these models for even larger populations, mainly because, and this sounds like a stupid Zen riddle, but no activity pattern at the level of hundreds of neurons, when you look at the detailed spiking pattern, will appear twice in your experiment. This is just the scale of the problem is so big that you don't cross the same river twice, right? So any notion of trying to make sense of what happens to particular patterns, or trying to look for this zip flaw thing that I described to you before, is not going to scale up. And so we were faced with the question of, if we wanted to make sense of what's going on here, so how could we, and how maybe the brain could try and learn to deal with these codes? And so we ended up with mixing ideas from 3 different places. So one is the kind of things we already done, kind of saying, well, we look at these models. It seems like there's a small set of things that seem to be governing what's going on. But so there's a sparse set of dependencies. We just need to figure out what they are. What I haven't shown you, but I'll just tell you that there's some notions of maybe there's some hierarchical organization in these structures. And if you wanted to think about what the brain might be doing, what should we copy about the ideas of dealing with these kind of things from the brain? So we know there's a lot of sparse and, at least to some approximation, random connectivity between things. We know there's a lot of cascades of linear and nonlinear terms. And so maybe that's something, kind of a hint of what we would like to use. And from machine learning, we know that if you live or you want to look at things that live in high-dimensional spaces, this whole field of compressed sensing is all focused on the fact that if you take things that live in really high-dimensional spaces, but their real dimensionality is relatively low, if you take a lot of random projections of these things, you can get very far in terms of describing what's going on very accurately, right? And so this goes back to the word of Lindenstrauss here many years ago, up the hill. So we try and see if we can put all of these things together, and we want to use maximum entropy formalism because that's kind of a nice way to try and find minimal models. And so here's the idea. So instead of building models that go with first order, second order, third order, which is very focused on the number of fingers that we have, maybe we should do something completely different. So we said, let's pick random functions. So if I think about my population, I'm going to pick random set of neurons, and I'm going to give them random weights. So this will be the 50 shades of green that I'm going to show you here. And so now I'm going to take this random group of neurons. I'm going to make it relatively sparse for reasons I'll show you in a bit. And then I'm going to compute some nonlinear function over this group of neurons. So in particular, I can try many different things. This is the one that I'll talk about most today, which will say, so each of the neuron xi will get some random weight. This will be this alpha I term over here. And this will be some nonlinear function g. And the thing I'm going to compute about subgroups of the population is this functions h mu. Okay? So I'm going to take a lot of these, and I'm going to say, just like I built the minimal model that relied only on the pairs or then on triplets, I'm going to build the minimal model that's consistent with the value of these random functions, that we'll call them random projections, where I'll pick threshold, strong threshold for the nonlinearity. So basically, I have small groups of neurons that I give them random weights, and I'm going to compute some set of these things. And I'm going to solve the same problem from before. I'm going to look for the minimal model that's consistent with the average values of each of these random projections and see what I get. So technically, this would mean that the probability that we'll denote as RP for random projections over the population will be now an exponential distribution. Well, I need to learn one coefficient lambda mu for each of these functions. And so just to give you the notion of what this means, it thinks that if you think of the populations, I plotted here these ellipsoids to try and describe, I picked some random group of cells, I give them some random weights, and that's kind of, in a sense, trying to think about covering the whole space with these random functions. Okay? No questions. So we're going to try this on data we got from the cortex. So this comes from recordings from the lab of Rusbekiani at NYU. And so there'll be prefrontal data from monkeys performing classification over moving dots, and recordings from Tony Mofshan's lab of V1, V2 neurons, and monkeys presented with drifting gratings. And we'll ask how well this model works and compare it to the other models. So the top line will be the data from the visual cortex, the bottom line will be the prefrontal cortex, and the columns will be, again, this independent model, the pairwise model, and this new random projection models. And again, every dot is one binary word, if you will, in this code. The x-axis is how often you see it, and the y-axis is the agreement between the model and the data in these cases. So again, the independent model sucks. The pairwise model is doing better, but not as well. And these random projection models do remarkably well in terms of describing what's going on. So again, this is with this random set of projections that we put in. So what's kind of nice about this, of course, so also this does really well in predicting third order and fourth order correlations in terms of how well things behave. If we ask, how does it capture other statistics of the code? So this is now the activity. So this is kind of looking at the synchrony over the population. So in the V1, V2 data, there were 170 neurons. In the prefrontal cortex, 160 something. And so this is now just collecting over the whole experiments in 20 millisecond bins, in each bin how many neurons were active. And this is kind of what we call the histogram of the synchrony values. You see the data in black, the independent model prediction in gray, the pairwise model in green, and you see basically these random projection models are within error bars from the actual data. So this is how accurate these models are. What's really nice about them is that the number of projections that we need is surprisingly small. So this is now measuring the likelihood of the model. So as a function of the number of projections, for reference, is the value of the pairwise model in green, some variant of it that adds higher order terms in blue. And this random projection model is a function of the number of projections that we have here. Again, we're talking about 170 neurons here. So if you had all the pairwise terms, this would imply nearly 20,000 parameters. Basically, by the time I have a few thousand of them, I do as well as the other model. And that's kind of the nice thing about these random projections, because there's no scaling issue of picking new ones. You can just keep on adding more and more of these random functions until you kind of get a good description of what's going on. But as we see here, it turns out that you actually can do really well with a surprisingly small number of these random projections. What I want to emphasize, because it's hard to see here, is that these orange blobs here are kind of designate what happens when we did this overlearning the models. And so this is kind of error bars over 100 different choices of random functions. So it's not that I had to be particularly lucky or particularly structured. Any random choice of these functions seem to be super robust in terms of the behavior of the models. I just need to get the right kind of coverage over what the population is doing as a whole. Questions?
There's a lot of questions. First, you did it here only on evoked responses, right?
Yes.
Do you know that it behaves similarly when there's no cue, like
yes.
spontaneous
So
activity?
yes. So if we look at spontaneous activity, which I'm not showing you here, then the models work perfectly well. The issue about the relation of the activity in the ongoing activity or spontaneous activity in the induced one is a different talk. So I don't have slides, but we can actually talk about the relation.
The same.
Yeah. The models work perfectly for all these different conditions.
And do you restrict the size, your functions that you are, the weights that you are generating?
Yeah.
Do you have some rules to the size of the pattern you are?
I'm not sure I said.
Because you showed the set of neurons, and then you selected a set of black or green neurons
Yes.
with different shapes. So this
So
number of red dots here is.
Oh, so I'll talk about the number in a minute, but my key point is I'm choosing them like this. And I want to do this as randomly as possible, which is part of what I want to try and push.
So there's no rules for this.
Yeah, I'm trying to be as ignorant and random and stupid as possible. It's my motto in life. Jonathan?
In different conditions, you have different parameters?
In different conditions for the...
Experimental conditions for the same network.
You mean in terms of the number of projections? Or
No, in terms of the parameters.
So I'll show you in a bit. Basically, it's if you have different population sizes, then you probably need different number of projections. But in terms of the ways we're going to pick these random functions, this seems to be super robust over many, many different things we try. I'll show you some more of that in a minute.
Yes. I mean, conceptually, how does that work out then? Because we know that, let's say when the subject is doing one task versus another, the correlation structure between neurons changes, they live in different subspaces, so on and so forth. So how does, what Elad asked, how does that add up then? How would the spontaneous, the same model would fit the spontaneous activity versus the task learned activity?
So what I'm telling you is that the set of random bases for which I'm going to learn the probability distribution, that works for many different kinds of stimuli. If you want to look at the detailed spiking patterns when you see a dog, when you see a cat, and when you sleep, then the details of the distribution are going to be different. And so the coefficient for each of these random projections would have to be different. What is super robust is that I can just pick one set of these random set of functions, and with that I can describe all of the distributions that you're going to see. Okay, so
even when even during the baseline versus when the monkey is looking at the moving dots? Like these are very different contexts, right?
Sure. So, maybe I need to try and explain this in a different way. I'm trying to build the probability distribution over these very long patterns, and the question is, which kind of statistics do I need to describe what's going on? So the fact that if you build this model for if you want to model the response for pictures of cats and pictures of dogs and whatever, then for each of these classes you might need different sets of lambdas here. But these functions H, you can use the same set and you learn everything with the same set of random functions.
I see, but the lambdas would...
Lambda will be different, yeah, which I'll get to in a bit.
Do you remove and you want to go file it?
No, I don't discriminate based on
So
firing
you're
rate.
very quiet human.
Yes. Whatever I need, that's what I'm trying to do. Okay. So, where were we? So, I'm sure this did not escape maybe all of you or many of you, but if I think about the thinking about the activity of these populations, and I'm trying to describe the probability of observing particular patterns, then again, I would need a particular set of lambdas for one kind of stimuli set and maybe another one for another set of stimuli, but these basic functions are enough to describe what's going on. It's something that you can immediately translate to a relatively simple neural circuit model. What is going on? Okay, that's weird. Sorry. Okay, so what you see on the left is basically a neural circuit that would implement this very model. So if these are the neurons I want to describe their activity, these random projections would be neurons that get random weights from the input layer, if you will. Each of these coefficients here will be randomly selected. These are the random green values I gave before. And in order to learn a model, all I need to do is to learn these weights that will combine these projection neurons together. And so basically I'll get a readout neuron who will basically compute the log likelihood of the probability. So there's a fairly simple neural circuit where all I need to do is to randomly pick the connections at this level here. And again, I do it like this. And in order to learn the probability over the particular set of stimuli, I need to just learn this set of weights. So there's basically one layer of weights that I need to learn. And this works under many different conditions. One of the really nice ones we obviously ask is how sparse do you want to be or need to be when you kind of work with these functions. So this is for these two different data sets and different population sizes. What I'm plotting is the likelihood of the model over test data, so measuring how good of a model do you get as the function of the sparseness. So we get both for V1, V2, and for the PFC data that there seems to be something of a sweet spot between 3 and 7 neurons in terms of the sparseness of these models. So we do really, really well. This is kind of naturally sparse in terms of how well we do. If you ask yourself, how much data do we need to train the model? So I'm plotting here the performance of the models as a function of the number of samples that we use to train it. And so for reference, this is the pairwise model in green, and its sibling, the k-pairwise in blue. We need many, many thousands of samples in these models to get accurate behavior. With these random functions, we can do fairly well with a few hundred samples. So we can get an accurate model for the activity, again, for a particular set of class of stimuli, that if I look now at the response of the neurons in prefrontal cortex during these tasks of classifying dots, we can do this at the level of getting a few hundreds or maybe a few thousand samples, which is as accurate as running the experiment again. And the reason is that estimating these random functions would be just because we compute whether they cross some threshold or not requires relatively small amounts of data. So this model works with surprisingly small amount of training data. Elon.
I'm not sure if I followed you the whole way, but the temporal issue, the time, how it affects.
So at the moment, I'm just looking at the sequence of patterns. I'll get to the temporal one in a little bit. Okay. We kind of thought that the fact that we have a simple circuit that seems to be implementing these kind of that could implement these kind of models, and things are sparse. So can we think about this as a real neural circuit of sorts? And so we said, can we come up with a learning rule that could allow us to actually learn these models? And so, because unlike the usual notion of asking about credit assignments or the issue of what's going on here in terms of the layers, because we're picking this set of weights to be random and we fix it, and all I need to learn is one set of layers here, this becomes a much easier problem than any notion of backprop or something like that. And so what we came up with is a trick that we stole from an elegant paper by Yashar Sol-Dixon and company. And I'm just going to give you the general idea because this is kind of nice but not that nice, is that if you think about how to try and learn these weights here, and you think that the activity of the neurons is then followed by some noisy versions of it, of this some kind of a noisy echo of what you just saw, that would be enough to give you a way to try and learn these weights relatively well. And this is the performance of this seemingly biological sensible way of trying to learn things just from the noisy instantiations of patterns or what happens right after that you've seen them. And so one last thing that we did in terms of thinking of how biological this version of the model might be is to say, well, and this kind of goes back maybe to the question that Jonathan asked before, what about neurons that don't fire that much, or maybe projections that don't do that much? So we said, what if we were to prune or throw away projections that don't seem to be doing much for us, and try to replace them with better ones? So what we did is we learn the model, we then throw away projections that seem to have a very, very weak coefficient. And so if we try and look at how well we do, it turns out that we can do even better with even fewer projections. So this is now, again, looking at these data sets over hundreds of almost 200 neurons. This is what happens for the model without pruning. If we allow ourselves pruning and replacing things with new random projections, we can actually get to the order of a few hundred projections that we actually described there. And this also results in changes in the activity and correlation, which I'll skip for now. So basically what we get is a shallow neural network, because we're not deep people at all, that relies on random connectivity, so you don't need to learn anything about the structure in advance. And it needs a surprisingly small amount of training data. It relies on sparse connectivity. It's super scalable, because if you need more projections, I'll just add more of them, and I'll show you this for larger data sets in a bit. Pruning, which is something we see in real biological circuits, makes it even more accurate. And kind of on the side, it also seems to be decorrelating inputs and things like that. So this is what we get from this. And we also have a learning rule that we can talk about later if you're interested. Now, in a sense, what this model is doing is computing the likelihood of its own inputs. That's kind of what the model that we're trying to do in describing the probability of observing particular patterns is built. And if there is such a circuit, you kind of say, well, why would the neural circuit in the brain want to compute the likelihood of its input? And then this now gets exactly to the question that Elad asked before, what happens if you have different stimuli? So let's say that I wanted to learn a model for describing the code of the population when there's pictures of dogs, and there's one where there's pictures of cats. So I get the green model on the left here and the red model on the right. So I can basically learn these two kinds of random projection models to describe the vocabulary of the system in response to each of these different data sets. Because I can do everything with random functions, which I picked like this, I can basically use the exact same projections in the middle. So I don't need two separate models. I can use the same set of projections and just have different readout for the same set of neurons. this will basically give me a way to do Bayesian computations with this kind of models that compute the likelihood on their own inputs. Okay? So we were kind of excited about this, and then we asked, the fact that so many of the biological features were nothing, or biological-like features were not things we put into the model, we asked, can we do even better? So, and kind of thinking about how these models are built, one of the obvious questions, partly due to referee number 3, was, you have these random functions that you pick. What if you played with the functions in some way, you try to optimize them in some way? And so we came up with a different version of this, which we call reshaped random projections. And so the idea here is now, if I want to describe the population, rather than what we did before is to say, I give you some set of random functions, which here I denote as f, and I need to learn how to mix them. What if I just give you the random functions and you're stuck with this set of weights, but you can allow yourself to change the functions? So graphically, this would mean that the random projections mean you have a random set of weights that you pick once and you're stuck with it, and you only learn how to combine them. This reshaped version is the version when you say, I take this random connectivity, and then I'm allowed to tweak the numbers on this connectivity. I'm not allowed to add more connections, but I'm stuck with the connectivity that I have, but I'm allowed basically to change the synaptic connections between them, where this thing is fixed and I cannot touch it. And for reference, we can do backprop on the whole thing and ask what happens if you try to train both of these things together. What we get is that all these models do really well, but you can already see from this scale of showing individual patterns that the orange and purple are doing much better than this naive random projection version. And to quantify this, we can now compute the likelihood of the models as a function of the number of projections. And again, with a surprisingly small number of these projections, we can now describe, again, this is the data set of our 178 neurons from the prefrontal cortex of the monkeys, basically as well as if we were to do backprop. And what's one really nice thing about this is that you can also try and look at what it does to the neurons, and it seems like doing this reshaping things kind of has an effect on the firing rates of the neurons. And so we kind of went back to all ideas of this notion of what happens to firing rate, and go back to Gina Turrigiano's work on homeostatic regulation of firing rates that was first shown in cultures and then in the awake active cortex. And these ideas of canonical computation by normalization from Carandini and David Heeger, and kind of say, what if we try to add these things to our model? So we have our random projection models. We have this reshaped projection models where we change the projections. And now I add two seemingly sensible biological features. One would say I'm bounding the kind of synaptic connections, how strong they might be. These will be these red models here that will be bounded. And the other is this normalized or homeostatic version of following Gina Turrigiano, and kind of ask what happens if you learn the models in this particular way. So this weird plot describes the performance of the model as a function of the kind of synaptic weights you allow yourself to use. Because once you have this homeostatic regulation or bounded value, you need to ask how much synaptic juice I put into the model. And what we find is that these homeostatic models are super robust to different values of the synaptic cost, and we can actually do things very well with very, very sparse and weak synapses. So this is performance as a function of the strength of the synaptic connections. The orange and the green points are just references of where the other models are. And these bounded models suck. That's not doing us anything. But this normalization thing is doing a really, really good job. And then we can also go further and ask, if you look at these homeostatic models and look at their performance, do we see that they actually, by this normalization, they would do homeostatic regulation? And what we find, if we look over training of these models, I'll just give you the summary here in the bottom right. Each of these crosses describes what happens to these different kinds of models. And basically, the tighter the regulation you have over the overall synaptic weights, just normalizing them, you end up regulating the firing rate to go back to be narrow in a kind of very tight regime. So these models not only do a better job of describing the data with fewer projections and weaker synapses, they also regulate the firing rates of the neurons very well. So we can go even one step further and kind of ask about, what's the sparseness of these models give us if you think about the biological structure of things. So we actually tried synthetic models where we generate data from a model that has a particular connectivity, and then we ask, how well would you do if you try to learn that with a different kind of random connectivity versus doing full backprop models. Basically, with random connectivity, you again can do really, really well compared to the other thing. And the last thing I want to say about this is that if you ask yourself what happens when you do this homeostatic regulation mechanism in the learning, is that you basically describe the overall vocabulary of the system by a random set of these projections that if you think about the space of activity of neurons, it's as if you had some kind of a hyperplane that separates these two things. And this reshaping process means that you kind of changing the projections that you use. And so we ask, what happens if you had different kinds of homeostatic regulation mechanism? What if you regulated synaptic inputs? What if you regulated synaptic outputs? What if you regulated both? We basically get the fact that they all perform similarly well in terms of giving very accurate models, and the notion of how they rotate these projections is not that different from one another. So this seems to be doing a remarkably good job of describing what's going on here.
Do you use any relation to the excitation inhibition balance, EI balance? So you don't use any of these?
So I don't need to take that into account at all.
There is no inhibitory neurons here.
There are. I just ignore their identity altogether.
You ignore the connections that they have.
I don't ignore the connection that they have. I'm building something that has, I'm using my random set of functions as a way to try and learn what the population is doing. If some of these neurons are inhibitory, some are excitatory, that will work just as well. I'm completely agnostic about the identity of these neurons. So in that sense,
yeah, I understand what you're saying, but I don't understand how it works.
If you take a balanced network and try to use this network to get
balanced network in the population.
Yeah.
Sorry, in the activity of the population. We tried and it worked really well. I'll show you in a bit.
So this maybe relate to that, but by learning these models, can you say anything about the encoding mechanism? Or the underlying latent variable?
So in a sense, I think of these random projections as defining the latent space that controls what's going on. Because this is a model that relies on all these small groups of cells, something between 3 and 7 in these kinds of cortical populations. And so if, rather than telling you, here's the full correlation matrix between all pairs that you need to put into account, what I'm telling you is that if you pick a relatively small set of random functions of these kind, these are the latent factors that seem to be controlling the population to a degree which is as accurate as running the experiment again. So predicting which patterns you'll get, we can do that remarkably well.
No, but this is not really how it was. What does the underlying neuroactivity encode?
How it's encoded in the real brain, I don't know. I'm just telling you that this is...
The question, does it tell you something about the dimensionality? Some rules, can you use the results, maybe not a specific weight, for example?
Yeah, because...
The dimensionality of the result random features.
So I have 178 neurons here. I'll go larger in a bit. There's 2 to the 178 possible patterns. I'm telling you that with a few hundred random functions like this, you can think of as the latent factor that control what's going on. I can build a model that is indistinguishable from the data. So in terms of the dimensionality of data, I'm giving you an upper bound on what you need because there might be an even better way to do this. But this gives you a surprisingly small set, I think, of latent factors that control the accuracy of the code to the degree that I predict, individual pattern frequency as well as the experiment. Ellie.
Like you say, the latent factors.
Well, sorry, a set of so this is a set of latent factors, and in fact, with the fact that when I pick a different set of random functions, I get similar behavior. This basically says that there's redundancy between them. So I don't know what there's. According to this, there's no the set of there is a. It's this way of doing this is so powerful that a random choice of these sets will do well enough. Is there an even smaller minimal set which is the magical one? Maybe, but
then following the unease that the questions here reflect, what did you learn about the brain? What can you do interesting? So, okay, so you describe this set of patterns, but then what? What's the next stage in terms of?
So if I can compute the likelihood of inputs for a particular input that accurately, I can now get a new pattern and ask what's the likelihood of the thing I just saw outside, right? So the fact that, one thing is for us to be able to compute to have an accurate model of the encoding of a neural population. The question of if you believe this circuit in terms of something that the brain might do, this gives the brain a way to compute the likelihood of the activity of the
That's fine, but the thing that interests me, for example, is I have 100 stimuli that I use to test the system,
Yeah.
and I want to know its response to stimulus number 101.
Mm-hmm.
Now, according to you, this would require estimating some new weights. Okay? Can you? What can I do in order to do this?
So let me skip this thing about scaling up to a few hundred neurons and use something else here. Sorry. So what we can do with this is to say, let's take now, this is an example of the kind of thing you might think about. If I think about 6 different points along an experiment, and I can look at what happens when, during fixation or target presentation or motion of the dots or delay, or so, I can now learn one of these models for each of these different segments. I need a surprisingly small number of a few hundred samples to learn a model for each of them. I can use the same set of latent factors to describe what's going on, which is a hint of what biology might be doing. And I can use these models to do the exact kind of classification and prediction of, if you now give me a new set of activity patterns, I can now ask how well I can actually decode what was shown or which segment this came from, which is what we can actually do with this. Okay? So the power of this thing, and sorry? 8 minutes. 8 minutes, okay. So we can do all of this also for temporal patterns. I want to try and maybe give you a snippet of why, where this thing, so actually, sorry. So to try and kind of get to the point of what you can do with these models, it kind of says, going back to Eddie's question, what I'd like to do is to get a new pattern and know something about what it means, right? Or how well I can decode it. And so this notion of trying, the fact that I have these models, what I'll try and argue, gives me a way to try and learn something about the semantic organizations of these patterns and figure out how they're organized. So if I think about the similarity of spike trains, this is the old paper from Jonathan Victor and Purpura, who kind of said, well, if I have one spike train up here in A and another spike train in B, I can think of the minimal set of changes I need to make between one to another to ask to define a metric or a similarity measure between different spike trends. But that's kind of a weird notion of how to measure similarity between patterns. What we did instead is to say, let's take now groups of 20 cells. This is something we did for the retina, but we're doing now for cortical data as well. And now let's take two kinds of movies. The things that's going to be in purple will be artificial white noise stimuli, and the things in green will be natural movies that we showed. And we're going to repeat them hundreds of times. And so what I can do now is that for every point in time, now that I have hundreds of samples, I can try and learn a model for the joint activity in this particular point in the movie. Okay? So for every point in the movie, I'm going to learn its own model. Because what I'd like to do next is to talk about which kinds of parts of the movie were similar to one another, or something about the structure of these patterns. So this is now the summary of what these 20 neurons did over 600 and something repeats of this particular point in the movie. So you see these are variations on a theme, so there's something there, but we're trying to make sense of what does it really mean. So what we do is we learn one of these encoding models of over the population. We tried this with pairwise model. We tried this also with these random projections. And so now what we want to do is we want to be able to, from these models, because they're so accurate, I can try and ask, can I learn something about the semantic similarity of patterns? And so I argue that the way to do this, rather than looking for a metric the way Victor did, is to say, if I think about two binary activity patterns, R1 and R2, that Ellie might be recording in the experiment, the measure of how similar they are for what the brain might do with them is to ask, what is the probability distribution over stimuli that might have elicited these particular patterns? So I want to measure the similarity of two neural activity patterns based on the distribution over stimuli they came with. And I'm going to measure this by the overlap of these two distributions. So if two binary words like this would appear once when you show one kind of stimuli and the other one where you showed a different stimulus, then these stimuli are similar because they kind of result in the same set of pattern would be encoding both. If these distributions are very different, then these patterns obviously encode very different kind of stimuli. And if you do that, then the reason we can estimate these distributions is exactly using these encoding models. So when we do that over the thousands of patterns that we get in the experiment for 20 neurons, this is now a map of the similarity of activity patterns if you order them by the firing rate in each of these population activity patterns. So there's 4,000 patterns in the Gaussian stimulus and a few more thousand in the natural movie. If you just measure which patterns seem to be encoding similar stimuli, and you order this matrix just by the firing rate. But if you cluster it, there seems to be a very clear structure of which patterns encode similar things in the stimulus. And the way to get this is exactly because we have these kind of accurate models for which we can ask for each of these different responses, what is the thing that's shared or common between them? So the power of these models, even if these latent factors are not necessarily the ones that the brain controls, is that it gives us a way to identify the population encoding noise, if you will, that defines the semantic meaning of each of these things. And then we can actually use these patterns to actually decode the movies. And what's kind of really nice about them is that if you now take a new set of stimuli and you take the activity patterns that you get and you ask, from these models, can we identify how many different modes of encoding do we actually get? We can compute for each of these patterns the kind of the canonical model it might be coming from, and we can do a fairly accurate decoding of what's going on with only a few hundred samples or modes of that we learn. So in terms of the structure of the code, there's some low dimensional, again, for this kind of population, we need only a few hundred modes of activity that encode what's going on. So let me summarize. And so in terms of what I tried to show you is that we have a new class of models that we can actually use to capture the joint activity of very large populations of neurons. It's super accurate. It's scalable. I haven't shown you the slides because time ran out, but we can actually doing this now for populations of nearly 1,000 neurons, again at the resolution of individual patterns and over temporal patterns. We have a kind of a candidate neural circuit that could implement this in the brain, which will allow neural circuit to compute the likelihood of their own input, which I would argue is exactly the thing that when you're trying to decode neural activity, when you're doing an experiment, this is exactly what the brain would be doing. And again, this is super scalable, and we have a way to use them to learn metric. And the thing about the structure of these networks, we'll wait for a different talk. And I just want to recognize the people who did all of this work. So what I've shown you today is mostly the work of Ori Maoz, who's now designing self-driving cars, and Udi Karpas, who's designing large language models, Adam Haber, who designs new medicines, and Elad Ganmor, who designs new ways to get you to buy things from commercials. So there's some pattern developing here, and Yoni Mazel, this is part of his PhD. Gashpar Tkachik and Ruz Bekiani were the main collaborators in crime here, and this is the people who gave us money. And thank you for your time. Yes?
So if I got you right, the optimal number of neurons per random projection like is between 3 to 7. How does that number scale with the number of neurons in the total network?
Yeah, so when we tried this up to 1,000 neurons, things that go up to between 7 and 10 works similarly well. There's a similar, or not similar, kind of interesting work from Ashok Plitu and Kumar in trying to look at the random projection patterns both in the factory bulb and in the factory tract of flies, and then in cerebral data, and they also come up with something between 3 and 5 as a magical number for these random patterns. So this is the number that seems to be consistent for these kind of populations. Yes?
Very interesting talk, thank you. So you showed that this random projection clearly improves the predictive power, but shouldn't this also help you predict noise or shuffled data? And then another question I have is, in the beginning you were talking about higher order correlations, which are multiplicative, but in the neural network model, isn't everything addition?
So I'm not sure I got the first thing about the modeling of the noise and the. Can you repeat that part?
So if these random projections have really good predictive power,
yes.
How do you know that that you wouldn't also do a better job predicting noise?
I'm trying to describe the activity. I mean, I'm learning these models for some training data, and then I predict on test data that I didn't train on. So, in terms of, you're asking, maybe it's it predicts things that shouldn't appear, or kind of I imagine things that are not there. So this would mean that if I sample these things, I can actually anchor myself against the experimental data, and I'll see, or I generate things from the model, and it will generate patterns that don't appear. We actually control for that. We see that, again, this is very similar to the data. We haven't seen place where it hallucinates, if you will, things that are not there. For the second part, part of why we went for these random functions, which are relatively simple in terms of their linear and nonlinear terms, is that doing the multiplicative things on higher order terms seems both expensive and not clear how to do this biologically. This is basically a set of random perceptrons, and that gives me the right vocabulary to describe populations to a high degree of accuracy. The higher order magic is hiding under the nonlinearity of the perceptron.
What happens to these random projections during learning? If you now learn a completely new thing, concept,
so
would you need only lambdas, or you need?
Yeah. So there's two versions of this. One is in terms of me building my model, and if you want to believe that or imagine that maybe there's a biological network that would do this. for the first version, I can do this with just changing the lambdas. For the biological version, it seems like if I do allow both the learning of just the lambdas or the projections themselves, and I only allow this renormalization of the kind that Gina Torrigiano sees, then I'll do even better. So part of what I think is kind of elegant here, again in this version where I just pick one set of random functions and I only need 2 ways to read them, is I can do this just with changes of synaptic connections in one layer. So this could work basically just with so I can basically just learn here and have, if I wanted to learn something else, I just need a new readout neuron and get its connections here. So this one learned to identify dogs. This one, if you now want to identify cats, you just need to learn this set of weights here. You don't need to rewire everything. It's, in fact, what this gives you is this catalog or corpus on which you can build your way to describe anything that you want. If you want 120 different stimuli, all you need is one set of random projections and then 120 different readout neurons. Each of them will get you the probability of which patterns will appear for this particular thing. So in that sense, it's as cheap as it can possibly get. These are the real latent factors that the brain uses. Someone needs to do the experiment. Thank you for your time.
פשוט יש לנו איזה מאמר של בן אנגלר, אתה זוכר מי זה? היה לי סטודנט כזה? ואני יש לי הרגשה שיש שם דיבור עם הדברים האלה, למרות שזה בדיוק הפוך. זאת אומרת, כל הגישה קצת הפוכה. האינפוט הוא מה שמשנה את התכונות של ה-projections, כאילו, של הרשת. אז מאחר וזה לא יתפרסם, אני אשלח לך רק זה יתפרסם ב-BioArchive. נורא מעניין אותי לחשוב מה אתה חושב על זה. בסדר?
לא סיפקתי אותך. לא. We seem to be doing well for, I'll just give a rough number. For 100 neurons, I need a few thousand samples and build up various samples that will repeat the thing. So, for example, if I have a lecture with sound of 30 seconds, I would like to repeat the 30 seconds. No, sorry. If you want to distinguish between one set of natural languages, no, sure, but tell me what are the different things you want, how many different things you want to calculate? So you have two different sounds. Okay. So now, if you repeat each of these sounds a few hundred times, over tens of neurons, the hypothesis is very
How does it compare to, let's say, if I do like independent analysis of my data?
that sense. So, to do ICA, you need to work relatively hard to try. Not that you can do ICL, but this doesn't really tell you what will be the other patterns that you might see. The thing is, this model is a generative model, so I can basically, it's an encoding model of what is going on, so I can use a sampling model. Right? So, correct. So I can use that to, so in a sense, let me say the thing I didn't say in the talk, which is usually so much in the same way. If you look at what you recorded from the neurons, sponsor the stimulus, and you split your data too, and you ask about the similarity. The model I'll build on the responses of the population in the first half will be a better predictor of what happens in the second half of the data than the matching of the two data sets. This is how I predict. So the model assigns probability to each of the patterns you will see. Very accurately.
Right, because for every stimulus, essentially you can, your model is a constraint, or after which there's a maximum entropy model, which means you're constraining it.
Use that word for a second, because I think this really. We have two stimuli. Right. I learn the probability of the response of the neurons to the first stimulus. Correct. And then a model for the other stimulus over the same set of neurons. So now I can, if you give me now a new pattern, I can now find out the probability of this distribution. That's my ability to encode which stimulus you just showed.
Right, and this encoding would be like, you can prove that this is better than, let's say, normally would like SVN or some sort of encoding. So if I give you a test pattern, ask you the stimulus it came from, yours would be the most robust way of doing the decoding.
So what we've done, and this is with and then kind of as a control, we asked them, what if we didn't use this? What if I give
לא, מה זה ספרנות? אבל גם בסיס קדמות יש לזה
So if you have enough neurons, you can do better. The benefit of these models is that the thing is going on these kinds of dependency are built exactly so you can code.
למה? אני אומר, בסוף אני צריך בערך 100 פונקציות כדי...
So this is the thing I want to explore. This thing creates some kind of representation that I can do really well with a simple linear thing that compares to So this, I think this is creating a linear representation that even it could have been, or could be, created a particular representation of what's and then whoever needs to be code needs to be very strong to try to make this right. The other option is to say, so the system has built a representation that is so robust that even this scenario is not very strong, but it's rather like LDA, or it will still do a decent job. I mean, I'll do better with this, but I think this is because it creates this kind of correlation between the neurons and the decision measure there. It builds a representation that is highly coded. So if you wanted to get perfect, then this is the best you do better. But in fact, you can do, I think this is creating representations that are relatively easy.
So do you think that I can look into this layer and probably learn something outside the exemption? Because essentially what you have is parameters which are these nodes. And so if in this particular space, like people would look into, can I get a nice stimulus representation? Yes. Because a stupid decoder can read out means that they are linearly separate. Right? So whatever non-linearity is my
But I'll tell you how to do, which I thought might make you so, these are cells in the CA.
Right.
So, we're kind of asking where the place comes from. What if, rather than, what's the natural each of them is computing the random function on landmarks in the corners of the room. And this will be a random set of projections. And then I make these neurons do some kind of thing. So this power of random connectivity, which I think is super powerful, I mean, I didn't get to talk about this in electronics stuff. We see that you can get very far with very small set of designs. Having random connectivity means that you don't need to specify anything except for saying you need to make 7 sandwiches. Find whoever you want. I don't care. Which is kind of what we're doing. I don't specify which kind of connect to whom. Just throw 7 of them, find someone, this will be. So, this is win control. I'm just saying, this notion of saying, does this create some kind of representation that you can try and make sense of?
Awesome. Once I get so maybe once I start primary features thanks so much.
Hey.
לא, מה זה לתאר? זה לחזור וללמוד ולעשות לו.
לא, אז התשובה היא כי אתה צריך לפרקשן פונקשן?
מאוד פשוט ללמוד את הסביב.
אם אני מסתכל על ההתפלגות,
אחרי זה הוא למד, זה שוטר שלו, הוא למד את הסביב. עשיתי ישיבות על הנוירונים, לא?
מה אתה מדבר?
רגע, כאילו יש לו איזה מין סקטרון אחר כך שלמדתי את הלוג לייטס. זה מה שהוא לא מצליח ללמוד ישירות מעל. לא, זה ספרס את זה.
הלמידה של
זה בדיוק אגב קצת פעם קצת ולו על תסמר ספיישן גדול.
ולכן ניסינו גם את החוק למידה ביולוגיה.
גם שם זה כאן גם. נראה לי שזה לקרוא. זה פעם שאלה, מג'יק. אני לא יודעת.
עכשיו נראה שיש מתוך ה-1,000 יש 10
הרבה פחות.
כל פעם אנחנו עוברים 5 רבנים גמרית, אז כנראה שנכון הרבנים יש איזשהו
בעבודה נגיד של השוק.
אני צריך לקבל איזשהו coverage של זה שאף נאמן לא יהיה מחוץ למשחק.
וגם שם הוא דוגם רנדומית כאילו?
כן, שם זה מודל של נירון, זה כאילו מודל אנקודים, לא דיקודים. השאלה היא איך זה אנקודים. שם יש very close connection from most fibers. sparse. יש expansion פי 100 או פי 100 פי. ואז יש רעיון שמעי זה אמור להיות איזשהו נוירון שקורא. לא זוכר מה שמבין. אבל הם מראים שהמספר המג'יקל נאמבר הזה בין 3 ל-7 זה יוצא מתוך קומבינטורית. של כמה כאלה אתה צריך כדי להעביר את כל המידע.
רוב הפרוטקשן שאני אייצר באופן כללי יהיו
אחר כך זה מידע, 1, 2, 3 זה מספיק, אחר כך מאיזה 7 זה כבר הכל, ואז כמעט מלא של שכלום.
אני אבוא אליך רגע, שרוצים...
היום הרגשתי אפילו, זה היה מעניין שהחלק הזה צבאי נפוט ואיכשהו. אבל כן, אז זה היה נורא מבין אחד.
אז הגעתי אלייך.
כן, לא, זה גם, כאילו שוב מבחינתי התודה היא על המקום שכזה מעבר לרעיון לעשות את זה בעיניי. כאילו, חושב באמת יש משהו בלמד איך לעשות מדע שהוא... אז תודה. אני מרגישה באמת שלמדתי המון, הרבה מעבר מהתיאוריה.
תודה רבה. מה הכי טובה שיכולתי לקבל?
כן, מורגש. תודה.
רוצה לקפה?
בטח, וגם לנסות להסביר את מה שלא הסברתי מספיק טוב. אבל לפחות עכשיו אני יודע איך אני צריך להסביר חלק מהדברים. כל הקטע של האנקודינג והדיקודינג, שמחתי יותר מדי על זה ברור ולא עשיתי את הדבר.
לא, אולי, מה, מבחינת המוטיבציה שלך?
לא, שזה שאני בעצם, אני חושב על זה שאני יכול ללמוד מודל כזה לכל גיבוי, או קלאס של גיבויים שאני רוצה. והעמדה של מה אני יכול לעשות אם אני רוצה לעשות דיקודינג, עכשיו, כמו איכשהו הכי לשאול. זה בעצם מה שלילה עכשיו עד השאלה הראשונה שלך, והיה לי שאלה אחר כך, ולא הסברתי מספיק טוב. בוא נתחיל.
יש סיכוי שפנים במעבדה יש רק חלב סוער.
אספרסו זה כל מה שאני.
מה?
אספרסו זה כל מה שאני.